In this video, I am going to discuss how cell division performs in mitosis. This is a diagram of mitosis. Interphase. This is the longest phase of the cell cycle, known as the resting phase. This is important for cell division because it allows the cell to grow, replicate its DNA, and make final preparations for cell division. Prophase. This is the first stage of mitosis, during which the chromosomes are condensed and attached to the mitotic spindle. Early prophase. The chromonymata, or the coiled central thread of a chromatid, still appears as one, although it starts to thicken and elongate. Middle prophase. Each chromosome visibly doubles and chromatids continue to thicken and shorten. Spindle fibers from the centriole spread and become longer. The nucleus becomes smaller and nuclear membrane starts to disintegrate. Late prophase. Chromosomes condensed, the nuclear membrane fades, and along with it, the nucleolus. Prometaphase. This is not a major stage of mitosis as it occurs briefly before metaphase. The chromosomes, led by their centromeres, migrate to the equatorial plane in the midline of the cell, at right angles to the axis formed by the centrosomes. This region of the mitotic spindle is known as the metaphase plate. Metaphase. This is the stage of chromosome alignment at the metaphase plate or equator. Early metaphase. At this stage, the chromosomes orient themselves along the metaphase plate. The centrioles have reached the opposite end of the cell. Late metaphase. The chromosomes arrange themselves along the metaphase plate with the centromeres occupying a central portion in the equatorial plate and attached to the spindle fibers. Anaphase. This is the stage of chromosome separation and migration to the opposite poles. Early anaphase. Chromatid pairs split or separate from each other through the pulling force of the spindles and begin to migrate to the opposite poles. Each chromatid is now considered as an individual chromosomes. Late anaphase. The chromosomes form two distinct groups at each pole. Each chromosome finds its way into each daughter cell, giving each cell an identical complement of chromosomes. Telophase. This is the stage of nuclear reconstitution, the start of cytoplasmic division. Early telophase. Chromosomes have reached the opposite poles and lie close to each other, forming a clump. Late telophase. The nuclear membrane reforms around each group of chromosomes. The nucleolus appears in each developing cell. Spindle fibers disappear. Chromosomes begin to uncoil just like how they were during interphase. Kinesis. This refers to division of cytoplasm of a plant of animal cell into two. The cell divides and two new daughter cells form.